All right, so again, here we're going to be talking about the different specific passive transport mechanisms. All right, we can divide it up into two different types of passive transport mechanisms. Now, remember that this is going to require no ATP or no energy, and we're moving from high concentration to low concentration. Simple diffusion is just going to be where we're going to move molecules directly across that membrane. No carrier, no channels required, no pore. It just goes directly through the membrane. All right, and again, we're using that concentration gradient. This is where we're going to allow for the transport of small molecules, nonpolar, or uncharged molecules, as well as lipid soluble substances. Okay, that'll be like gases and certain lipid soluble drugs. Okay, now we can divide it up into two different types. We have electrolytes and non-electrolyte substances. All right, if we are transporting non-electrolyte substances, this is going to be driven by that concentration gradient. Okay, and the rate of that's going to depend on three things: the permeability of the membrane, so how permeable that membrane is to that specific non-electrolyte substance, the surface area of that cell, and then the concentration difference. All right, so the difference between the concentration of that substance on the outside of the cell and on the inside of the cell. Obviously, the bigger the difference, the bigger the concentration difference, the more likely it's going to want to go through that cell or through the cell membrane. Okay, these principles are based off of what we say Fick's law of diffusion. Right, and then we also have electrolytes. Again, this is driven by concentration gradient, but we're also going to add another element where we're going to also drive it by way of an electrical gradient. We call that an electrochemical gradient. So it's going to be based on both the concentration as well as the charge. Okay, so an example of this would be the diffusion of sodium through that sodium channel. All right, and then we have facilitated diffusion. Facilitated diffusion means that it's going to need a helper. Okay, it has a helper protein, a carrier protein, or a channel protein. Again, no ATP is going to be required. We're moving from high concentration to low concentration or down the concentration gradient, same as simple diffusion. All right, but we have to have this channel or this carrier that helps. In this case, transport's going to be a lot faster than simple diffusion. All right, but the problem here is that it can become saturated. If you don't have enough carriers, if you don't have enough channels, then that will be the fastest it can go through. All right, because you have to have the availability of those proteins in order to transport these materials. All right, so channel mediated is going to be where we kind of just have uh, this protein that allows us to have a pore or a hole that goes from one side of the membrane to the other. Okay, molecules are going to be able to pass through that hole from one side to the other. Okay, this is often going to be selective for specific ions like sodium, potassium, chloride, or small polar molecules. All right, small molecules that have charge. We also have carrier-mediated proteins, and these aren't as simple as the channel-mediated. The substance won't just go from one side to the other through a hole in the membrane. What has to happen here is that that substance has to bind to a binding site on the carrier, or it has to signal in some way for that carrier to change its shape. That carrier protein changes its shape in order to transport that molecule across the membrane. Okay, this is going to work for larger molecules as well as like glucose or amino acids.